So I wish to thank the organizers for giving me the opportunity to present our study. As we heard before today, autism is diagnosed using behavioral assessments such as ADOS, these current methods, behavioral method to diagnose autism, potentially neglect the biological variation, which may reflect the vast heterogeneity and comorbidities seen in autism. Therefore, taking into consideration these biological factors can pave the way for more precision therapy. Urine metabolites provide a valuable insight into how the body biochemical processes operate and how they utilize key components from a diet together with the gut microbiome. Unlike blood and cerebrospinal fluid, CSF, which have overall consistent composition under normal conditions, urine composition may vary during the time. Since urine compounds vary, it is easier to track changes, making urine as an ideal source for biomarkers for complex brain disorders. There are several studies reporting the different patterns of metabolites in the urine of children with autism. This information has been collected from several countries, including China, Italy, France, demonstrating that these findings are not specific to a unique region or can be explained by cultural factors. In an Italian research, the researchers also reported correlation between urine measurements and behavioral assessments, which supporting the evidence of the connection between urine metabolites profiles and behavioral assessment. Among the urine metabolites, we can differentiate two distinct groups: the internal metabolites, the non-food metabolites, endogenously produced in the body and serve as part of the biochemical processes such as Krebs cycle. The external metabolites, which are the food or the exposome metabolites, are exclusively originated from food and are subsequently processed by the body. The question of this study was whether we can find a way to use the metabolites of the urine in order to classify between autism and control, neurotypical children. The first step was, in every machine learning study, was to collect the data. In this study, we collected 293 polar urine metabolites and from Israeli cohort of 32 children diagnosed with ASD and 20 age-matched neurotypical children. Among the urine metabolites, 189 were internal metabolites and 104, the rest were external metabolites. The next step in machine learning study is to train the data. It is important in such complex problems of studying biological issues like trying to classify ASD from control based on metabolites to use different machine learning classifiers. The random forest classifier is a very strong, and we will see in the edition of in the next slide, it's a very strong classifier that can be applied to many different tasks. However, using single decision tree is faster and much easier to understand. Logistic regression was also used in this study. It's very interpretable results and can deal good with multi-dimensional data. While naive-based classifier is very efficient, however, it has very strong conceptions on independence between the features. The combination of several algorithms combines each algorithm limitation and strengths and can give us a better perspective of the results. The first step was test the data. The test method used here was tenfold test, which tests building the model with 9/10 of the data and testing it with the rest 1/10. This is done 10 times, each time with a different portion of the data. Results are shown here. Here, we used all the metabolites for the classification, and the random forest performed best. Actually, I can say random forest performed best in all the rest of the cases we will see here. And it achieves 85% accuracy and area under curve of 0.9. The most distinguishing features are listed here. It is evident that only glycerate has significance. Sorry, this is the laser. Oops, back. Okay. Only glycerate has a significance difference between control and neurotypical. However, combining several features gives us a better classification results. Here, it's the classification based on only 189 internal metabolites. This seems to be similar to the results with all metabolites, a little lower, 81% successful accuracy and AUC of 0.86. Also, the two most distinguishing features are similar with the all metabolites. However, the three, the N-acetylcysteine, proline, glycine, mesotitrat, appeared only in the internal analysis. The results of classification based on only external metabolites surprisingly was very low and achieves only 71% accuracy and AUC with 0.72. Interestingly, none of the most distinguishing features from the external metabolites didn't appear in the list of the all metabolites classification. Here, there is the combination of all the results of the three metabolites classifiers. On the right, you can see the area under curve. On the left, it's the accuracy. Colored blue are all metabolites. Color orange, internal metabolites. And color green, as we see before, it's the external metabolites. So why it was surprised that the results of the external metabolite is so low? Because children with autism usually suffer from eating disorders and may have very similar diet and restriction that might help distinguish them from neurotypical children. However, we can see here that the results of the classification based on external metabolite is rather low compared with internal metabolite and external and all metabolites. Having established the metabolic signature patterns, we next investigated whether these results correspond with the biochemical findings. And we used two tests, the ATEC, Autism Treatment Evaluation Checklist, and SDQ. This analysis is very crucial to understand the interaction between the results of classification, the current classification behavioral assessment and metabolic assessment. So the data was collected from parent questionnaires, the parent field questionnaires, for both behavioral classifiers. It is important to mention, I guess you know, that ATEC is more specific to autism, while SDQ is more general and oriented to mental health diagnostic. Random forest, again, was the best classifiers of the answers for the ATEC questions. It is important to notice, I guess, that the questions were filled by the parents of the same cohort children that we did the analysis of the metabolic classification. It is not surprising that the ATEC questions can distinguish between ASD and control. However, using this classification, finding the most distinguishing features, we can say that there are several features more abundant, more important to indicate the results than other behaviors of the children. The results of classification based on the SDQ question is listed here. Again, very successful, not surprising. We can see here the most significant features. Asterisk means that we find negative behavior in the children with ASD. For example, happiness level was low in children with ASD. Combining everything together, this is the detailed results of all our classifiers, almost all. I take out the external. We have here a heat map, or maybe it's more precise to say a table. Each line in the table presents a child in our cohort. Each row is a different classifier. The most right column is all the metabolite classifier. The second is internal classifier, internal metabolite classifier. Then we have the ATEC and SDQ classifiers. Color green means actual ASD is predicted as ASD, which is the true positive. Light green means actual control was predicted as control. This is the true negatives. The red means actual ASD predicted as control. This is a false negative, and the pink is the false positive. Actual control was predicted as ASD. Okay, thank you. It is interesting to see that some cases, not many, several cases, where the metabolites, where the behavioral classifiers were not able to predict ASD, the metabolites did. However, we still have many false negatives here in the metabolites classification. However, still we can see the combination of both approaches as a more complete classification and can provide us a more precise therapeutic for children with ASD. To summarize, we demonstrated that using exclusively polar urine metabolites without any behavioral information. We were able to classify children with ASD from neurotypical with 85% accuracy and 0.9 AUC. Interestingly, internal non-food metabolites differentiate ASD better than external metabolites. Our metabolite analysis may provide a complementary biological marker that corresponds with behavioral patterns. The behavioral assessment is still the cornerstone of diagnosing autism, but sometimes it can be challenged because of variation, different comorbidities, heterogeneity, and adding another classification feature can assist to have a better result. So the future plan is to use this classification in order to have a more sophisticated diagnosed autism and also personalized treatment. I wish to thank my collaborators, Dr. Yael and Dr. Maya from a New Start company. My gratitude also go to Professor Ivan Elliott from Bar Ilan University, the head of the biobank, to Julie Carmel, the project coordinator and the clinical trail coordinator, and to Maxim and Dr. Maxim and Dr. Sergey, which conduct the analytical, the analysis of the urine in order to break down into metabolites. And I wish to thank you for listening.
Thank you very much, Shula.